I'd like to thank Affymetrix for the opportunity to come and talk. After the first two talks, which I found really enlightening about genetics, I should warn you I'm not a geneticist or a statistical geneticist, but an epidemiologist. And if you are really interested in the details of the genetics relating to UK Biobank, please do come to the meeting at 1 o'clock here in this room, because there will be people involved in that meeting who do know about genetics, Peter Donnelly and Martin Tobin, who can talk about the way in which the genetics data in UK Biobank can be used and how they worked with Affymetrix to design the chip. So what I wanted to really talk to you about is the experiment of UK Biobank, because the funders, who are essentially the UK government, the Medical Research Council being the equivalent of the NIH, and the Wellcome Trust, a major charity, decided to set up this resource as a resource for any researcher anywhere in the world to use for any kind of research that was in health-related research that was in the public interest. So the experiment is, how do we make it available? How do we persuade people that there are no catches, that this is their resource? Our job is to build it, to enhance it, to make it easier to use, and for other people to use. And I'm finding that a very exciting and very interesting experiment. So what is the cohort? So 500,000 British men and women aged 40 to 69 who were registered with the National Health Service in the UK, so that we were able to, first of all, identify them, to invite them to join the study, and then in the long term to link into all of their health-related records to find out what happens to them. They were recruited between 2006 and 2010. And we have general consent for all types of health-related research, for both academic and commercial research use, and for follow-up through all health-related records. So it is there to do really whatever you want to do with it, provided it's health-related research in the public interest. The Medical Research Council and the Wellcome Trust, for reasons that escape me, decided that they were going to put a very large amount of money into this cohort. I mean, I can see why it's a good idea, but I'm amazed that they could see it was a good idea without having to put in a grant application. I got a phone call while on holiday in Italy saying, would you take on the job of setting up UK Biobank? You've got a budget of $100 million. That's a pretty nice phone call to get. And they've doubled that investment in building this resource. It's allowed us to get really detailed information about the participants. So there was a lot of discussion before UK Biobank started about, was this the way to go forward? Should one, if one was interested in genetics, do a prospective cohort? Probably not. It's not very cost effective. But if you're interested in genetics and other risk factors, environment, lifestyle, and their interactions, then in the long run, a prospective cohort is a very cost-effective way of looking at the interaction between lots of different risk factors and lots of different diseases. The ability to look at pharmacogenomics, to look at lifestyle effects and how they interact with genes. So the debate was, okay, we're going to do a prospective cohort. How detailed should it be versus how big should it be? And there was a big debate in the UK about, should it be big and dumb, or should it be smaller and clever? Should it be deep in the phenotyping? And I think what we've been able to do is actually do both. We started out thinking, well, how do we take the funding we've got and get 500,000 people? What's the most we can get with that resource? How can we be cost effective in using that to get the most information? And one of our watchwords was defer. Don't do what we don't have to do now. Wait until later in the hope that it might become cheaper or that the funders might give us more money. What are the things we have to do now? And so we focused on that. We particularly focused on demonstrating that it was doable, because again, there was a lot of debate. Could you really recruit 500,000 people? Once we demonstrated that it was doable, that we could get detailed questions and measurements and samples, we went back to the funders and said, we'd like to get even more information. So in the end, we ended up with big and smart, with deep phenotyping in substantial subsets, and I want to describe that. But before we started, we got a lot of working groups from around the UK and international input, considering different types of exposures, different types of disease they would want to study, and what were all the questions or measurements or samples that they would want to collect. And as you could imagine, one ended up with a very large number of questions. It probably would have taken a day of interviews if you had done all of them. So what we did was get those groups to kind of work together and see where there were overlaps, and got it down to a set of questions that would still take a long time with interview and would cost a lot of money. So to get around that, the IT team built a touchscreen questionnaire that allowed us to get a lot of questions answered by participants at very low cost, get automatic coding, and went beyond questions. So on average, participants spent about 50 minutes answering a whole series of questions, and you can see the amounts of time on different kinds of questions, to give you a sense of the range of different things that we were asking about. And the beauty of the touchscreen was it was very cheap. We had one member of staff would look after perhaps about 15 or 20 people answering questions rather than a face-to-face interviewer. It was private. So you didn't have to worry, could someone hear you when you answered the question, how many sexual partners have you had? So people could skip questions, but they didn't because they felt it was private. So we were able to ask, if you like, very intrusive questions, to get a lot of detail from these participants. And the touchscreen allowed us to go beyond questions and into tests. So cognitive function tests, reaction time, various kinds of executive memory tests, hearing tests. So it was a very effective way of getting a lot of information. It could also then be used to trigger particular questions of particular participants given their answers during a more limited interview around a particular medical history and medication, which we could enhance by linking to the health records that go back in time before people joined the study. We were aware there were deficiencies in the kinds of information we could ask. So food frequency questionnaires on this touchscreen are not a very precise way of finding out about people's diet. Asking people about their exercise and activity is a problem because, of course, they lie. They say they do far more than they really do. But one gets part of the way there, and then one can think about other things one can add. In terms of physical measurements, when we did the maths, this is what we could afford on all 500,000 people. A whole set of kind of standard, obvious things. Blood pressure, heart rate, height, waist, hip circumference, weight, body impedance, spirometry, heel ultrasound to look at bone density. But again, the IT team made sure that the data were coming straight out of the machines into the database. So we don't just have, say, FEV1, FVC, we have the whole spirometry curve. The IT was ensuring that the quality of the blows was good, so that if they weren't, people had to repeat it. So there was quality control built in to the collection of all of the data. And as I mentioned, when things were running smoothly, we got another $10 million to add a series of other measures, some of which are shown here. I mentioned the hearing test, a measure of vascular reactivity. Moorfields Hospital in London, it specializes in eye disease. They pulled together a group that thought about, well, what would we like to know about vision? So visual acuity, refractive index, intraocular pressure, retinal images, and that was when I learnt what optical coherence tomography is, which is essentially, if that's your retinal photograph, this is your OCT, you can see the layers through the retina, you can see the nerve fiber thickness, which has been associated with cognitive function. So we've now got about 85,000 of these, and the eye group are the group that have really understood the concept of UK Biobank. So if you go and look up UK Biobank I Consortium, you'll find that they have decided it's their resource, and they have all these groups around the UK and internationally that are focusing on particular areas of eye disease. So they don't think it's our resource, it's theirs, and they are now analysing the imaging data and starting to publish. And then to try to get at fitness, an ergometry test done in about 85,000 people. And those data are currently being analysed so they can go into the database. The critical thing, though, was also what biological samples. And so we worked with a lot of groups, and this was coordinated by Tim Peakman, the Deputy Chief Executive of UK Biobank, a lot of groups to say, well, how should we collect the samples? Not in order to do assays now, but in order that assays that you might want to do, or that you might be able to afford to do in 5, 10, 15, 20 years' time, can be done on the samples in the way they're collected. And so in the International Journal of Epidemiology, a monograph was published of all of the pilot studies on the different kinds of assays that can be done, the impact in terms of stability in the different samples. Of course, one gets things wrong. We didn't realize that the amounts of DNA that we would need in the future would be much less than we thought. So we have samples that have been stored in such a way that one can immortalize the cells and produce more DNA, probably never be used for that. But in fact, we've done some work to repurpose, and we found that those samples could be used for other things. But we have about 55 mils of blood, so 3 tablespoonfuls of blood, in a range of different tubes, a urine sample, a saliva sample, that is available for research. Again, to address this issue of how do we make the resource smarter, how do we phenotype the participants in greater detail, we had a working group that kind of started to throw problems at us. What were the additional things that we'd really like to know about the participants, or indeed about their disease outcomes that we might not be able to get from linkage through health records? So they focused on enhancing the characterization. I mentioned the problem about food frequency, so a group in the Cancer Epidemiology Unit in Oxford, working with the Medical Research Council Unit on Nutrition in Cambridge, developed an internet diet diary. We pinged out an email to the 250,000 participants for whom we have email addresses, saying, what did you eat yesterday? They go through the link, they tell us in detail what they ate yesterday. A few months later, we ping it out again, what did you eat yesterday? So we're building up a diet diary. For those of you who've done work in nutrition, diet diaries on paper are very problematic in that they take a long time to analyze. You actually have to train people to complete them, participants, and then you have to spend a lot of time actually analyzing them. These, very easy to complete and automatically coded, and the data go into the database in terms of nutrients based on asking people exactly what they ate. Not so much about characterizing the participants, but characterizing their outcomes. We've most recently sent out a web-based questionnaire looking at the cognitive assessment again since their baseline visit and adding additional assessment of cognitive function. And this is something we'll repeat over time to try to identify people who are becoming cognitively impaired. As I said, if you ask people about activity, at least in the UK, they lie. And so we've now got 100,000 of the participants who've worn an accelerometer on their wrist for a week. And we have now analyzed those data, turned it into energy measures, and this month we will be releasing the data on 85,000 of those for researchers to use. Coming to the bit I guess you guys are interested in, the genotyping. 500,000 participants were genotyped. It was an extraordinary experience, actually. So we had a meeting in about November 2012. Peter Donnelly, who will be speaking later today, and a whole group of people from the UK and the US were there. What should we be doing in terms of genetics? Should we genotype all of them? if we could have the money. Should we sequence some of them? And the consensus was very clear. Genotype as many of them as you can. This was about November or so of 2012. We discussed it with our scientific advisory board. The funders heard this. They thought this is a good idea. And then suddenly, the financial year was looming. And the National Institute of Health Research, which is the Department of Health funder for research, said, we've got this money that will disappear if we don't spend it by the end of the financial year. I think this was about January. So by the end of March. And they said, genotyping sounds like a good idea. So we had 2 months to spend quite a large amount of money. We had to tender, we had to identify who to work with, and that's where we ended up working with Affymetrix. We talked with a lot of people, Neil Reisch particularly, who's the president of the association, said he'd had a fantastic experience with Affymetrix. Peter Donnelly talked with all of the different potential groups. And I think the key thing that swung it for him, so he chaired our group, and he can talk about this at lunchtime, was the amount of input in terms of the design. We had a very short time window to make a decision, and it was the ability to get a custom design with a lot of input from Affymetrix that I think made a huge difference. And so we actually started extracting DNA in November of 2013. Sam Murphy in our group in Manchester coordinated that and completed the whole process, including all the genotyping, within an 18-month period, which I think is pretty extraordinary. The data, in terms of the quality, I'm told by the Wellcome Trust Centre for Human Genetics in Oxford, again, Peter can talk to this later today, was very high quality. Colin Freeman there did the quality control. Jonathan Marchini did the imputation. And so this was the chip that ended up being used, a bespoke chip with this content, which means almost nothing to me. As I said, the DNA extraction and the genotyping was completed in about 18 months. And the measured genotypes and about 70 million imputed genotypes for the first 150,000 participants were released in May of 2015. All the genotyping is now complete. The data are being QC'd in Oxford. Then there will be re-imputation. I have no idea what number this will become when they re-impute. When they first told me they were going to do imputation, they said 20 million, then it became 30 million, then it became 70 million. So Jonathan Marchini hasn't told me what he's going to try to get it up to when we release the data in mid-2016. And a lot of researchers have got the data and are playing with it, which is exactly what it's for. We then went back to the funders again and said, we need to turn some of the sample, the depletable sample, into data. The problem is if we use up the sample on this little study here, this little study here, there, we won't get comparable data on particular risk factors. Some will measure cholesterol in one set with one measure, some will measure it with another measure, and they won't be comparable. For the kinds of analytes that lots of different people will want for lots of different research, it would be much better to do that on all 500,000 participants. And so this is now going on at the moment. We've pretty much completed the urine analysis and the HbA1c analyses, and these markers, cardiovascular markers, cancer markers, diabetes markers, renal markers, bone and joint, etc., will be released during 2016. And then finally, our enhancement working group tried to push us to the edge, and they said, what about doing some imaging? And so we said, okay, but it's a prospective cohort. So the problem with a prospective cohort is you have to do things in a lot of people because you don't know who will develop any particular condition. So that means if you want to image from the point of view of a prospective cohort, you have to image a lot of people. So we sent out an email to a lot of imaging experts around Europe saying, we've got this idea of imaging 100,000 participants in UK Biobank. And they wrote back and said, that's a really good idea, but there's a typo in your email. You meant 10,000. I mean, I'm not joking. And we said, no, we really meant 100,000. And it's taken about 5 years for them to understand why that's the case, and for them to really engage in developing protocols that could be done on that scale. So we built the first imaging center in Manchester with a 3 Tesla magnet for brain imaging, a 1.5 Tesla magnet for body and heart imaging. We're doing DEXA, so low power radiation of bone, joints, and body. The nice thing about the DEXA is it gives you automated analysis of body composition, but it's not as detailed as the MR of the body, but that currently is not very easily analyzed by automatic means. Ultrasound of the carotid arteries to look at atherosclerosis, and repeating the baseline assessment, more detailed cognitive functions, because we're interested in dementia, ECG to look at rhythm, and collecting biological samples as well. We've just actually gone past 6,000 people in that center. We're just waiting, based on our report of that pilot phase, to find out whether the funders will release the remaining funding to allow us to build 2 more centers and to image 100,000 participants. We have shown that it's doable, and we've shown that we get very high quality data that are useful for research. And I just wanted to illustrate one example of that. So body mass index, we've been hearing about how important it is as a determinant of risk. This is a meta-analysis of prospective cohorts, about 60 of them, in total about 1 million people followed for 12 years, that we published in The Lancet in 2009, looking at the association between body mass index and mortality from stroke or from heart disease, these log linear associations throughout the range down to a BMI of about 22, and then a kick up, which is probably causal, because if we look in similar associations in China, which we've done, where BMI is much lower, then this J shape is in the middle of the distribution, but it's at the same point. But here we've got BMI very strongly related with risk. But BMI is a really crude measure. You know, it's always said Tom Cruise has a fantastically high BMI, or probably did, because he's got a lot of muscle. So you know, you're not really getting a direct measure. You're getting a crude measure that is associated with the thing that you really want to measure about fat and about fat distribution. So here are 2 pictures from from Jimmy Bell at Hammersmith Hospital, who's on the imaging working group of UK Biobank. 2 individuals, same sex, similar age, same body mass index, same estimated body fat from impedance, but about a 3-fold difference in internal fat. Will that be importantly related to disease? Well, when we started out with UK Biobank, we didn't anticipate that we would be able to get automated analysis of the body MR data. But in fact, by generating data at scale, as we've seen in the genetic area, that drives technological advantage. And what's happened is it's done so with imaging. So there are groups that have now developed automated approaches to analyzing body MR, and they've done that on the first 5,000 people in UK Biobank. This shows the association between body mass index on the x-axis and subcutaneous adipose fat from the MR. A very high correlation with subcutaneous fat. But with visceral fat, there's a less good correlation. And with liver fat, an even worse correlation. So if BMI, with all of its deficiencies, is strongly related to risk, what might more direct measures of fat and fat distribution be in terms of relation to risk, and then improving our ability to assess gene-environment interactions? So finally, a prospective cohort, the key thing is in the prospective, how do we find out about the health outcomes? Well, the beauty is that we have a National Health Service in the UK, and we can link, as in many places, to death and cancer registries, which we are doing, to inpatient and outpatient hospital episodes and related procedure registries, so we get coded data on all of these, to the commercial system suppliers in the UK that provide the data handling for family practitioners. We're now linking into the primary care records to get at health conditions that don't get people into hospital, to prescriptions, to diagnostic tests. We can link to other health-related records, to disease registries, dispensing records, imaging data that's increasingly becoming put into central databases, to screening records. And of course, as I mentioned with the cognitive function, direct to participants, using web-based questionnaires to get at those kinds of health outcomes that may be underdiagnosed. So cognitive decline, mood disturbances, pain, disability, so that we can not only phenotype the participants, but phenotype the health outcomes in great detail. And we have to be able to do that at scale in two senses. We'll have very large numbers of individuals during follow-up, as well as prevalent at baseline, who we want to be able to ensure that we have well-validated health outcomes. And not only will we have large numbers of any particular condition, but there will be many, many conditions that different researchers will be interested in studying. So we need to scale in terms of range of diseases and number. So finally, the principles of access. Available, as I mentioned at the beginning, to everybody. There's no preferential or exclusive access. It does not involve collaboration with us. It is your resource. The costs are actually the costs of us extracting the data and giving it to you, or extracting the samples and giving it to do assays. And we're looking at ways in which we can turn imaging or samples into data, which are then even cheaper to give to you. Obviously, the biological samples, some of it at least, is limited and depletable, and we need to try to control that. And one of the ways we're trying to do that is by having phases of assays done by us so that we can then release the data. And the only requirement is that the findings go into the public domain and that they're returned so that others can look at them. Our website's pretty easy to remember, UK Biobank. And so what makes it special? Well, it's prospective, so it can look at the full effects of particular exposures on a wide range of different diseases, or vice versa. It is, as I hope I've persuaded you, very detailed in terms of the characterization of the participants and their outcomes. And the most obvious thing, of course, is it's big, which provides for substantial statistical power. So it's that unique combination of breadth and depth that I hope will persuade you to use it as a resource. We've had a lot of use from the UK. Increasingly now that the genetic data have become available, we're seeing increasing international use, particularly in the US. We've seen relatively little commercial use, but I'm hoping that will increase as well. So thank you very much for your attention, and I hope you will become users.